Imagine your body as, well, an incredibly sophisticated fortress. You've got those immediate alarms, the barricades, that's your innate immunity,
Yeah.
right? The general first responders.
Exactly, the quick reaction force.
But what happens when a really cunning invader, learns those basic defenses?
Hmm.
You need something more. A specialized, intelligent counterattack. A system that actually remembers past battles and tailors its response with just, well, astonishing precision.
And that's precisely where adaptive immunity comes into play. It's quite remarkable.
Welcome back to the Deep Dive, everyone. We're the place that takes complex info and turns it into, hopefully, a clear insights you can use.
That's the goal.
Today, we're doing a deep dive into adaptive immunity. We're using Chapter 7 of Understanding Pathophysiology as our guide here.
A really foundational chapter for understanding this system.
So our mission for you today is to unpack your body's third and most specialized line of defense. We'll look at how it builds that specific long-lasting memory against invaders.
Yeah, we'll trace the whole journey from the birth of these immune cells right through to the precision strikes of antibodies and T cells.
And even how this incredible system changes as we age. Get ready for, I think, a few aha moments.
Definitely.
Yeah.
It really is a marvel of biological engineering. Understanding it gives you such profound insights into how resilient the human body actually is.
Right.
As the chapter points out, adaptive immunity, sometimes called acquired immunity, it steps in when those initial barriers are breached.
Okay.
And the innate inflammatory response, well, it's already started its work. Think of innate as the first containment, buying time.
Buying time, okay.
While adaptive immunity slowly builds up those defenses and, crucially, prepares the body to remember and respond better to future threats.
So inflammation is the immediate sort of general alarm. What makes adaptive immunity so different? So adaptive.
Yeah, that's the core question, isn't it? There are a few key differences that really set it apart. First, it's inducible.
Inducible, meaning
Meaning it has to be switched on. Unlike innate immunity, which has components just, always ready, the specialized cells and proteins of adaptive immunity are produced specifically in response to an infection.
okay, so it takes time to build up.
Exactly, it develops more slowly. Second, it's exquisitely specific. While innate gives that general response, adaptive immunity is precisely tailored to the exact microbe causing the trouble.
Wow, so it's not just a general alert, it's like designing a custom key for one specific lock, not just trying a bunch of master keys.
That's the perfect analogy, exactly. Right. And third, its protective elements are long-lived and systemic. They provide lasting protection throughout your body, which is quite different from the more temporary effects of innate immunity.
Right.
But maybe its most remarkable feature, the one everyone knows, is memory.
Memory.
If you get reinfected with the same microbe later on, those protective cells, those antibodies, they get produced immediately, offering what can often be permanent, long-term protection.
That memory function is just so fundamental, isn't it? Explains vaccines, recurring infections.
Absolutely foundational.
But these two systems, innate and adaptive, they don't just work separately, do they? They must interact.
Oh, completely. The chapter really emphasizes this. Despite their different roles, they're highly interactive, complementary, you could say. They really can't function fully without each other.
How so?
Well, many parts of the innate system are actually needed to kickstart the adaptive responses. And then, in turn, the products of adaptive immunity, like antibodies, they often boost innate defenses. It's a collaboration.
Okay, makes sense. So let's dive into the key players then, get some vocabulary straight for this smart defense system. When we talk adaptive immunity, we're mainly looking at specialized cells called lymphocytes, T cells and B cells, and antibodies, those crucial proteins in your blood.
Right. And the targets that these antibodies and lymphocytes recognize, those are called antigens.
Antigens.
Antigens are usually molecular patterns. Think proteins, carbohydrates, maybe fats found on microbes, or infected cells, even abnormal cells like cancer cells.
Got it.
Now, here's a subtle point, but it's important. While all molecules that can bind to immune receptors are antigens, not all antigens are immunogens.
Hold on. Not all antigens are immunogens. Okay, break that down for us.
Sure. An immunogen is specifically a molecule that's capable of inducing an immune response, triggering it.
Right.
An antigen, just by definition, can bind with antibodies or lymphocyte receptors. But to actually cause a response, it usually needs to be, well, substantial enough. Large enough.
Okay.
For instance, there are these small molecules called haptens. They're antigens they can bind, but they're too small on their own to trigger a full immune response. They only become immunogenic able to actually cause that response after they combine with a larger carrier molecule, like one of our own proteins.
Okay, so that sounds like the classic poison ivy example. The urushiol oil itself is the hapten, the antigen.
Yeah,
But it only becomes an immunogen, triggering that awful rash when it binds to proteins in your skin. Then the body sees the whole combo as foreign.
that's a perfect real-world illustration.
Yeah.
So the adaptive immune response, it unfolds in two major phases, sequentially. First, the generation of clonal diversity, and then clonal selection.
Two big stages, building the arsenal, then activating it.
You got it.
Right.
These stages build and then deploy your body's specific immune weapons.
All right, let's explore that first phase, generation of clonal diversity, which is amazing because it happens before you even meet a specific foreign antigen.
It really is visionary, isn't it?
Yeah.
The purpose here is to create this huge population of B and T cells. Each one is uniquely equipped to recognize pretty much any foreign antigen you might ever encounter.
Any antigen. How many are we talking?
The estimate is the potential to recognize over 10 to the 8th. That's 100 million different antigens. It's staggering. And this process starts before birth, continues throughout life.
100 million. Wow. Where does all this incredible diversity get generated?
It happens in specialized primary lymphoid organs. For B lymphocytes, or B cells, that's the bone marrow, deep inside your bones.
Okay.
For T lymphocytes, or T cells, it's the thymus. That's a gland located just behind your sternum. These organs are like the basic training grounds.
Training grounds. I like that.
Yeah, where immature lymphoid stem cells mature and develop their unique recognition abilities. If you are mapping the immune system, these primary organs are the central hubs. They send out their trained cells to secondary lymphoid organs like lymph nodes, the spleen, which are positioned all over your body like outposts.
And how do they actually develop these unique recognition abilities? Let's start with B cells.
Okay, B cell development. In the bone marrow, lymphoid stem cells mature into B cells. A critical step is developing the B cell receptor, the BCR.
BCR.
Yeah. Picture the BCR like a complex structure right on the B cell's surface. It's basically a membrane-bound antibody, usually IgM or IgD type, linked to internal bits that send signals.
And they're all unique.
Exactly. This huge diversity, the ability to recognize millions of different things, comes from a process called somatic recombination. Segments of DNA within the developing B cell get literally shuffled and rearranged, so like genetic shuffling, to create a unique receptor for each single cell.
So each B cell gets its own unique genetic hand dealt for its receptor. What about T cells? Similar process.
Very similar idea, just in a different place. T cell development happens in the thymus. Lymphoid stem cells travel there and develop their unique T cell receptors, or TCRs.
TCRs.
Right. The TCR is also a complex structure, often described visually as having two distinct protein chains, alpha and beta usually, with variable regions, again, unique to each cell, and it's linked to a CD3 complex for signaling inside the cell.
Okay.
Like B cells, TCR diversity also comes from that same kind of gene shuffling, of that genetic rearrangement. And during this maturation in the thymus, T cells also start expressing another molecule on their surface, either CD4 or CD8.
CD4 or CD8, those sound important.
They are. They act like co-receptors, helping the T cell interact correctly. Those that keep CD4 become T helper, TH cells. Those with CD8 become P cytotoxic, TC cells.
So even at this early stage, we're seeing specialization, the helpers and the killers emerging.
Precise.
But wait a second, if these receptors are generated randomly, shuffling genes, what stops them from accidentally recognizing and attacking our own body? Our healthy tissues? That seems like a huge risk.
That is a critical point. It's called self-tolerance. And the body has a rigorous quality control system built into this development phase.
How does it work?
Basically, in the primary lymphoid organs, the bone marrow and thymus, any developing B or T cell that reacts strongly to self-antigens, components of our own body, is largely eliminated, destroyed.
Eliminated? How?
Through programmed cell death, apoptosis. This process is called clonal deletion or central tolerance. The chapter notes that over 90% of developing B cells and an incredible 95% of developing T cells are actually eliminated this way.
95%.
Yeah.
Wow. That's a massive amount of self-correction happening silently.
It is. It's absolutely essential to prevent autoimmune diseases, where the immune system mistakenly attacks the body. It's the body's crucial safeguard against itself.
So the result of this whole first phase, this clonal diversity generation, is a population of what the book calls immunocompetent but naive B and T cells.
Exactly. They're fully developed, ready to recognize an antigen, but they haven't actually encountered their specific one yet.
Right.
These naive cells then migrate out to those secondary lymphoid organs, lymph nodes, spleen, etc., And they circulate, basically waiting for their specific antigen to show up. Think of them as highly trained soldiers deployed to outposts, ready for the call to action.
Okay, poised and ready. Which sets the stage perfectly for phase 2, clonal selection.
Right. This is the phase triggered by actual exposure to a foreign antigen. It starts pretty much from birth and continues all through life. This is where a specific immune response against a particular invader really kicks off.
And most antigens, they can't just wander up to a lymphocyte and activate it, can they? They need like an introduction.
That's absolutely right. Most antigens need to be processed first and then presented to the immune cells. This vital job falls to specialized cells called antigen-presenting cells, or APCs.
APCs, okay. Who are they?
The main ones are dendritic cells, macrophages, and even B cells can act as APCs. They basically do one of two things. They either engulf antigens from outside the cell, those are called exogenous antigens,
Exogenous outside, got it.
or they process abnormal things from inside the cell, like viral proteins being made, or markers from a cancer cell. Those are endogenous antigens.
Endogenous inside, okay. And they present these processed bits using special molecular platforms, the MHC molecule?
Exactly. The major histocompatibility complex, MHC molecules. In humans, you'll also hear them called human leukocyte antigens, HLAs. Same thing.
HLAs, right.
They're glycoproteins found on the surface of most of your cells, except red blood cells, interestingly. And they come in two main types, or classes.
Class 1 and class 2.
Correct. MHC class I molecules are found on basically all your nucleated cells, so almost every cell in your body. They specialize in presenting those endogenous antigens, the inside job threats like viruses or cancer specifically, to T cytotoxic TC cells.
Okay, class I shows inside threats to the killer T cells. What about class 2?
MHC class II molecules are more specialized. They're found only on those professional antigen-presenting cells, dendritic cells, macrophages, B cells.
Ah, okay.
And they present exogenous antigens, the ones picked up from outside, specifically to T helper TH cells.
So it's a really specific delivery system. Class 1 TC cells for internal problems. Class 2 THE cells for external threats captured by APCs.
Precisely. If you could picture it, imagine the APC chewing up an external microbe, putting a piece on an MHC class II molecule like a flag, and showing it to a T helper cell.
Right.
Or an infected cell putting a viral protein flag on MHC class I for a T cytotoxic cell to see. It ensures the right response gets triggered.
It really is like a carefully orchestrated cellular symphony, as the chapter puts it. Let's talk about those interactions. Start with the T helper cells. They seem like the conductors.
They really are. T helper TH lymphocytes are absolutely central. They orchestrate pretty much the entire adaptive response, both the cellular and humoral arms.
How do they get activated?
An APC presents that processed antigen on its MHC class II molecule. The T cell's unique TCR recognizes that specific antigen flag, and its CD4 co-receptor molecule basically docks onto the MHC class II molecule itself.
Like a handshake.
More like a handshake plus a secret password. It's not just binding. It needs additional co-stimulatory signals between the APC and the T cell, plus chemical messengers called cytokines to get fully activated. It's a multi-step process to make sure it's a real threat.
Okay, so once they're properly activated, these TH cells then specialize further, directing traffic, basically.
Exactly. Depending on the chemical signals, the cytokines in the environment around them, activated T cells differentiate into specific subsets.
Like what?
Well, you get Th1 cells, which primarily help activate macrophages and cytotoxic T cells, driving that cell-mediated killing response. Then there are Th2 cells, which are crucial for activating B cells, helping them make antibodies driving the humoral response.
Okay, Th1 for cell killing, Th2 for antibodies. Any others?
Yes. There are also Th17 cells. They recruit other inflammatory cells, like neutrophils, and are important against certain types of bacteria and fungi. And then you have T-regulatory Treg cells.
Tregs. They sound important.
Hugely important. They act as the brakes. They limit and suppress the immune response, making sure it doesn't go on too long or get too intense. They're vital for maintaining that self-tolerance we talked about and preventing damage.
Wow, that's amazing strategic specialization. But the chapter mentions a really dangerous exception to all this specific recognition, something called superantigens.
Ah, yes, superantigens, SAGs. They are a critical and dangerous exception. These are toxins produced by certain microbes, like some strains of Staphylococcus aureus or Streptococcus pyogenes.
What do they do?
They basically cheat the system. They bypass the normal specific antigen recognition. Instead of fitting neatly into the antigen binding groove, they bind to the outside surfaces of the T-cell receptor and the MHC class II molecule, sort of clamping them together
Okay,
nonspecifically.
wait, what's the effect of that?
The effect is that they activate a huge number of T helper cells, maybe up to 20% of all T cells all at once, regardless of their antigen specificity.
That sounds bad.
It is. This massive nonspecific activation leads to an enormous release of cytokines, what's often called a cytokine storm. This causes widespread severe inflammation, fever, dangerously low blood pressure, shock. It could be fatal. It's a microbial tactic to overwhelm the host's defenses.
Chilling how a tiny molecule can cause such chaos. Okay, what about the actual assassins? The T-cytotoxic T-cell lymphocytes.
Rather, TC cells, your body's elite killers, they're specialized in finding and destroying cells that are compromised from within.
Like virus-infected cells or cancer cells?
Exactly. They recognize those endogenous antigens, the inside job flags, presented by MHC class I molecules on the surface of those abnormal cells. Their TCR binds the specific antigen, and their CD8 co-receptor docks onto MHC class I.
And then?
Once they're properly attached and activated, these effector TC cells trigger apoptosis programmed cell death in that target cell. They essentially tell the compromised cell to self-destruct, eliminating the source of the problem cleanly, like a targeted hit.
Okay, so that covers the killers. Now let's circle back to the B cells and how they get selected to become antibody factories. B-cell clonal selection?
Right, so those naive immunocompetent B cells circulating around, they have a cool trick. They can often bind directly to soluble antigens floating around using their B-cell receptor, BCR.
Without an APC?
Sometimes, yes, especially if the antigen has repeating units that can engage multiple BCRs at once, but often for a really strong response, especially for protein antigens, they need help. After binding the antigen, the B cell actually internalizes it, processes it, and then presents it on its own MHC class II molecules.
So the B cell becomes an APC itself.
Exactly. It presents the antigen it found to an already activated Th2 cell. That helper T cell then provides the necessary confirmation signals and releases specific cytokines, like interleukin-4.
And that's what triggers the B cell?
Yes. That interaction triggers the B cell to proliferate like crazy, making lots of copies of itself, and then differentiate into specialized plasma cells.
Plasma cells equals antibody factories.
Precisely. These plasma cells are dedicated to churning out huge amounts of soluble antibodies, all specific for that original antigen the B cell first encountered.
So T helper cells are usually crucial for getting the best antibody response.
For most antigens, particularly proteins, yes, they drive the high-affinity antibody production and memory formation. However, the chapter does mention T-cell independent antigens.
Right, what are those?
These are often large molecules with lots of repeating units, like some bacterial polysaccharides. They can sometimes directly activate B cells without any T cell help, just by binding to and cross-linking many BCRs simultaneously. This usually leads mainly to IgM antibody production, and typically less memory.
Okay. And the incredible result of all this clonal selection, both T and B cell, beyond making active fighters, is generating those crucial memory cells, right?
Absolutely essential. During clonal selection, both B cells and T cells differentiate not only into effector cells, plasma cells, TC cells, but also into long-lived memory cells.
And they just wait.
They remain largely inactive, circulating for years, sometimes decades, until, that is, they encounter the same antigen again.
And then?
Then they spring into action incredibly quickly. They offer a much faster, much stronger, and more effective immune response the second time around. This rapid, potent recall is the whole basis for immunity after infection, and of course why vaccination is so powerful.
Okay, that covers the cellular choreography beautifully. Now let's really focus on their weapons. The antibodies, the stars of humoral immunity.
Right. Antibodies, also called immunoglobulins, or IGs. These are the soluble proteins produced by those plasma cells we just talked about. And your body makes 5 main classes, each with slightly different structures and, importantly, different jobs. IgG, IgM, IgA, IgE, and IgD.
5 classes, and the chapter has diagrams showing their shapes. IgG is that classic Y shape, right? But IgM looks bigger, like several Ys joined together.
That's a great way to picture it. IgG is indeed Y-shaped. It's the most abundant antibody in your blood, making up about 80–85% of the total. It provides the bulk of your protective activity against many bacteria and viruses. And critically, it's the only antibody class that can cross the placenta.
Ah, so that's how moms pass immunity to babies before birth.
Exactly. Maternal IgG gives newborns vital passive protection for their first few months. IgM is physically larger, usually a pentamer like 5Y shapes joined together in a star. It's the first antibody class produced during that initial primary immune response. It's very good at activating complement.
Okay, IgG the workhorse, IgM the first responder. What about IgA? That one's special too, isn't it?
Very special. IgA is found in blood, yes, but its main claim to fame is as secretory, IgA, SCGA. This is the antibody found in your bodily secretions, tears, saliva, mucus, gut fluids, breast milk.
Guarding the gates, basically.
Precisely. It often forms a dimer, two Ys joined together, and has an extra bit called the secretory piece that protects it from being broken down in harsh environments like the gut. Its job is to stop pathogens from even attaching to your mucosal surfaces in the first place.
Crucial frontline defense.
Yeah.
And the last two, IgD and IgE.
IgD is found in very low levels in the blood. Its main known function is actually as that B-cell receptor, BCR, on the surface of naive B cells. IgE is also usually in very low concentrations, but it punches above its weight. It's specialized for two main things, triggering allergic responses and, very importantly, defending against parasitic infections like worms.
Parasites. How does IgE help there? We'll come back to that. But first, it's amazing B cells can switch the type of antibody they make, isn't it?
It is. It's called class switch recombination. A B cell that initially makes IgM, for instance, can later switch to making IgG or IgA or IgE. Critically, it keeps the same antigen specificity. The tips of the Ys still recognize the same target.
So it changes the antibody's function without changing its target.
Exactly, and this switch is directed by cytokines provided by T helper cells. It allows the immune system to tailor the antibody response, choosing the best tool, the best FC region function for the specific job or location, like switching from IgM for initial complement activation to IgG for longer-term protection optimization, or IgA for mucosal defense.
Okay, structure-wise, you mentioned the Y shape. How does that relate to function?
So think of the Y. The two arms at the top, those are the Fab fragments. Fab stands for fragment antigen binding. These are the variable parts that physically bind to the antigen. Each antibody has two identical Fab regions.
Okay, the arms grab the antigen. What about the stem of the Y?
That's the FC fragment. FC for fragment, crystalline, just how it was first isolated. This FC region is a constant part for each antibody class, like all IgG FC regions are similar. It dictates the antibody's biological function. It's the part that interacts with complement proteins or binds to FC receptors on phagocytes or NK cells.
Got it. Fab binds the target, FC determines the action, and the actual binding site on the antigen is the epitope, and the site on the antibody is the paratope.
Precisely. It's that specific key interlock fit between the epitope on the antigen and the paratope on the antibody's Fab region. Highly specific interaction.
So what do these antibodies actually do once they bind? What are their functions?
They protect you in several ways, categorized as direct and indirect mechanisms.
Okay, direct first.
Directly, they can physically interfere with the pathogen. Neutralization is a big one. The antibody binds to a virus or a toxin and prevents it from binding to your cells and causing harm. Think blocking the key from entering the lock.
Makes sense.
They can also cause agglutination, clumping together of particulate antigens, like bacteria making them easier targets, or precipitation, making soluble antigens clump together and fall out of solution.
Okay, neutralization, clumping. What about indirect actions?
Indirectly, they act by engaging other parts of the immune system, primarily through that FC region. A major one is complement activation. IgM is especially good at kicking off the complement cascade, and IgG can too. This leads to inflammation and direct lysis of microbes.
And the other big one is helping things get eaten, right? Opsonization.
Yes, opsonization. IgG is the main opsonizing antibody. When IgG coats a microbe, phagocytic cells like macrophages have FCC receptors that bind strongly to that IgG coating. It's like putting handles on the microbe, making it much easier for the phagocyte to grab and engulf it.
Antibodies as eat-me signals. Love it. The chapter also mentions monoclonal antibodies. These are huge in medicine now, aren't they?
Absolutely massive. Monoclonal antibodies are basically lab-produced antibodies that are all identical because they come from a single B-cell clone. This means they have uniform specificity and binding strength for one particular target epitope.
And that's useful because
you can design them to target exactly what you want. They're used in super specific diagnostic tests. Think pregnancy tests, tests for infections, and, incredibly importantly, in therapies. Targeting cancer cells, blocking inflammatory molecules in autoimmune disease.
Yeah.
It's revolutionized treatment for many conditions.
Okay, back to IgE. You said it helps fight parasites. How exactly does that work? The chapter has a figure on this.
Right. IgE plays a unique role against large parasites like helminths, worms. which are too big for phagocytes to just engulf.
Oh, what happens?
IgE antibodies produced against the parasite will bind to the parasite surface. But crucially, IgE also binds very strongly to FTIC receptors found on mast cells and basophils, immune cells located in tissues. Okay, when the parasite antigen then cross-links the IgE already bound to these mast cells, it triggers the mast cells to degranulate, releasing a whole host of inflammatory chemicals.
And these chemicals?
These chemicals attract other immune cells, especially eosinophils. Eosinophils are specialized granulocytes that can damage and kill large keratinocytes by releasing toxic proteins directly onto their surface. So IgE acts as the trigger, calling in the heavy artillery, the eosinophils.
Fascinating mechanism. A specific antibody for a specific kind of threat. And our bodies also have a whole separate system for surfaces, the secretory immune system.
Yes, often called the mucosal immune system. It's like a specialized defense force for all the external surfaces, linings of the gut, respiratory tract, eyes, mouth, urogenital tract. It operates somewhat independently.
And CG is the main antibody there.
Yes, exactly. Secretory IgA is the star player. It's actively transported into secretions like tears, saliva, mucus, breast milk. Its main job, as we mentioned, is to prevent pathogens from attaching to and invading through these mucosal barriers. It's like painting the surfaces with a non-stick coating.
And super important for newborns through breast milk.
Absolutely critical. Maternal colostrum, that first milk, is packed with SIGA, providing essential passive immunity, protecting the baby's gut from infections while their own immune system is still developing.
This leads perfectly into that fundamental concept showing memory in action. The primary versus secondary immune responses.
Right.
The chapter has a graph showing this clearly.
Yes, it's a classic illustration of immune memory.
Yeah.
The primary response is what happens the very first time your body encounters a specific antigen.
And it's slow?
Relatively slow. There's a lag phase, maybe 5 to 7 days, sometimes longer, while those initial clonal selection events happen, finding the right B and T cells, activating them, getting them to proliferate.
Okay.
During this primary response, IgM antibodies appear first, followed later by IgG. The total antibody level rises, peaks, and then gradually declines. Importantly, memory cells are generated.
But then the second time you encounter that same antigen...
It's a completely different ballgame. The secondary response is triggered by re-exposure to the same antigen, thanks to those memory B and T cells waiting in the wings.
Faster.
Much faster. The lag phase is significantly shorter, often just a day or two.
And stronger.
Much stronger. The amount of antibody produced is far greater than in the primary response. And crucially, the main antibody produced is IgG, which is generally more effective for long-term protection. This rapid, robust secondary response is why you usually don't get sick, or get much less sick, the second time you meet a pathogen, and it's the entire principle behind vaccination.
Fantastic. Okay, so that's humoral immunity covered. Let's switch back to the cells, cell-mediated immunity, and the direct actions of T lymphocytes.
Right. Cell-mediated immunity is all about the effector T cells doing their jobs directly or orchestrating others. We've talked about T helpers and memory T cells. The other key players here are the T cytotoxic TC cells, the direct killers.
assassin.
And also other cells influenced by T cells like macrophages, plus the natural killer NK cells, which have a unique role. And don't forget the T regulatory Treg cells keeping everything in balance.
So TC cells, remind us again, their main job.
Their main job is direct destruction of your own body cells that have become abnormal, primarily virus-infected cells or tumor cells. They recognize those endogenous antigen fragments presented on MHC class I, bind via their TCR and CD8, and then induce apoptosis in the target cell. Clean removal.
Got it. Now, natural killer NK cells, they sound similar, but the chapter says they're different. Lack antigen-specific receptors.
Exactly. NK cells are fascinating. They're lymphoid cells, but they're considered part of both innate and adaptive immunity. They don't have those rearranged, highly specific T cell receptors. Instead, they recognize cells based on general patterns of stress or abnormality.
Oh.
One key way is by looking for the absence of normal MHC class I molecules. Many viruses and some cancer cells try to hide from TC cells by deliberately removing MHC class I from their surface.
Trying to become invisible.
Right. But NK cells are wise to this trick. If an NK cell encounters a cell that lacks sufficient MHC class I, it interprets that as a danger signal and kills the cell. It's called missing self-recognition.
Clever. Anything else they do?
Yes. They can also kill target cells coated with IgG antibodies. They have FC receptors for IgG.
Oh.
When they bind to antibody-coated cells, they get activated to kill. This is called antibody-dependent cellular cytotoxicity, or ADCC.
So NK cells are versatile killers, working differently than TC cells. And other T cells act more like managers, sending signals.
You could say that. Different T helper subsets, as we discussed, release specific cytokines to direct other cells. Th1 cells release interferon gamma, which superactivates macrophages to become better killers, M1 type. Th2 cells release cytokines that activate macrophages for tissue repair and antiparasite responses, M2 type. Th17 cells release signals like IL-17 that recruit neutrophils and other phagocytes to sites of infection, especially bacterial or fungal.
So they tailor the response using chemical messengers. And finally, the T regulatory, TREG, lymphocytes, the peacemakers.
Absolutely, the peacemakers. Treg cells are crucial for putting the brakes on the immune response. They actively suppress the activation and function of other effector T cells. This is vital for maintaining peripheral tolerance, preventing immune responses against your own tissues out in the body, and also for shutting down responses once an infection is cleared, preventing chronic inflammation.
How do they suppress?
They use various mechanisms, including releasing immunosuppressive cytokines like TGF-beta and IL-10, which directly inhibit other immune cells. They are absolutely essential for preventing autoimmunity and keeping the whole powerful system from running amok.
This entire system is just incredible and so dynamic, learning, adapting. But it also changes across our lifespan, doesn't it? Let's touch quickly on age-related factors. How does it work in newborns?
Right. For pediatric considerations, newborns are immunologically quite immature. Their own adaptive immune system isn't fully up and running yet. They have relatively deficient antibody production on their own, and their phagocytic cells and complement system aren't as active as in adults.
So how are they protected?
Well, they can produce some IgM, especially if exposed to infection while still in utero, and they start making limited IgA soon after birth. Their own IgG production really only kicks in gradually over the first few months. The crucial protection comes from mom.
The IgG crossing the placenta.
Exactly. That maternal IgG transferred across the placenta provides vital passive immunity that protects the infant during those vulnerable first 6 months or so while their own system matures. It's a fantastic natural protection.
A head start for mom. Okay, and at the other end of life, geriatric considerations. Does immunity decline?
Generally, yes. Immune function tends to decline with age, a process sometimes called immunosenescence. We typically see diminished T-cell function. Older individuals often don't respond as strongly to new antigens, like in vaccinations.
Why is that?
A key factor is the thymus, that primary lymphoid organ where T cells mature. It involutes, meaning it shrinks and becomes much less active, starting around puberty and significantly reduced by middle age. This reduces the output of new, naive T cells.
So fewer fresh troops being trained.
Pretty much. We also tend to see reduced antibody responses, especially to novel antigens, and sometimes an increase in the levels of circulating autoantibodies, antibodies that mistakenly target self-tissues, even if they don't always cause overt disease.
Okay, wow. We've covered a huge amount. Let's try to recap the absolute key takeaways from this deep dive. What did we really learn about the body's ultimate defense system?
Well, first, adaptive immunity is your highly specific memory-driven protection. It's distinct from, but works closely with, the faster, more general innate defenses.
Right, and it starts with clonal diversity, making a massive library of unique B and T cells before they're needed. Then comes clonal selection, where specific cells get activated when their particular antigen shows up, usually presented by APCs using those MHC molecules.
Then we have the two arms. Humoral immunity is all about antibodies made by plasma cells, proteins like IgG, the main workhorse, crosses placenta, IgM, first responder, IgA, mucosal protector, IgE, allergies, parasites, each with unique structures and jobs. They neutralize, opsonize, activate, complement.
And cell-mediated immunity. That's led by T cells. You've got the TC cells, the cytotoxic killers destroying infected or tumor cells directly. You have the T8 cells, the helpers, the conductors directing B cells, TC cells, macrophages via cytokines. And don't forget NK cells, the innate-like killers catching cells hiding MHCI. And the crucial Treg cells keeping the peace and ensuring self-tolerance.
And critically, memory cells are formed in both arms, allowing that much faster, stronger secondary response. And we saw how this amazing system isn't static. It needs maternal help in newborns, passive immunity, and tends to decline somewhat as we age.
Okay, here's something to really think about. Consider this. Your adaptive immune system isn't just a defense mechanism, it's constantly learning. It's remembering, it's refining its strategy against this constantly changing world of pathogens we live in. What does that really mean? It means your body is essentially a dynamic, living library, a record of every immune battle it's ever fought. And it's capable of writing new chapters of defense with every single new encounter it has. It literally grows stronger and wiser through experience. How profound is that?
It truly is one of the most elegant and complex systems in biology.
Well, thank you so much for sharing the insights from the chapter and guiding us through this really fascinating deep dive into adaptive immunity today. We really hope you, our listeners, feel more informed and maybe even a bit awestruck by your own immune system.
Hope it was helpful.
On behalf of the entire The Deep Dive team, thank you for joining us.